Okay, so is this, okay, good. Sounds like this is on. So I really want to thank you for the invitation to come out here. This is the first time I've actually been out to UT. I've been to Bandera several times, but I've actually haven't been to the civilized part of this part, or the non-country western part, I guess, before. So I've had a great, I guess, morning and afternoon talking to a lot of different people doing all kinds of stuff. So I'm looking forward to sharing what our most recent work, I guess, over the last couple of years, and then, getting some feedback, and hopefully have some time for discussion at the end. So as Andrew said, we've been interested in mitochondria and their role in the aging, and specifically in skeletal muscle for several years. And over the last few years, we've had some results that I'll kind of share in the beginning that have really forced us to change the way we think about this dysfunction and how it might contribute to skeletal muscle pathology. So I'm going to kind of share that journey with you a little bit today. There'll be some published data, some data in process of being published, and then some really sort of preliminary kind of early stages stuff. So I'll get my disclosure out of the way. I'm a scientific consultant for Stealth Biotherapeutics, which is developing one of the compounds I'm going to talk about today. So this is just a brief outline of what I'm going to talk about. Just spend a few minutes giving a little bit of background on mitochondria and skeletal muscle and aging. I don't think I have to justify studying skeletal muscle too much for this group, but just in case. And then this will be the bulk of what I'll talk about in the middle here, is just this reversal of some of the age-related decline that we've observed by treating with the mitochondrially targeted compound. And then get into mechanisms a little bit, and then finally talk a little bit about the translational potential. So I think most of us probably know that the decline in skeletal muscle, or sarcopenia, which is loss of both mass and function, is really kind of one of the hallmarks of aging. You can really, basically you can tell an aging person by the structure of their muscle, basically, even someone that's well exercised. This is a cross-section, MRI cross-section through the quadricep of a 24-year-old and a 65-year-old female who are matched for BMI. So obviously, one of the things you see is a reduced muscle mass, but also this fatty infiltration decline in muscle quality, basically. And this is quantified here in this. I think this is. I don't know if this is. I don't think this was a review. I think it was an actually research paper out of the Mayo Clinic, showing this decline in muscle mass in men and women, and also this decline in strength. So this is kind of a regular part of the aging process. And sort of, why should we care about this? Again, I think most of us appreciate that. Sometimes, depending on the audience, you have to remind people that skeletal muscle it's not just to carry around the brain and the heart, but it actually plays an important role in overall health. And, one of the most obvious is loss of muscle function leads to exercise intolerance. And that can invoke an image of, people, their marathon time slowing down, or they're just, becoming a little less active. But this, taken to its extreme, is a loss of independence. And then associated with that is an increased risk of falls, increased disability with many neurological diseases, and then ultimately increased hospitalization and nursing home care. This important role of muscle function and exercise is highlighted in this review paper from, I guess, what's this, about 15 years ago, where they looked at risk of death or risk of mortality on this axis across multiple diseases. This is hypertension, COPD, diabetes, smoking, obesity, and cholesterol, and related that to the exercise tolerance of this group. So these dark bars are people with a high exercise tolerance, measured here in metabolic equivalence. And then these people are a low exercise tolerance. And you can see across many of these disorders, risk of death almost doubles as you go from a high exercise tolerance to low exercise tolerance. Now, you can argue about which way the causation arrow points here. There's some debate whether the lack of exercise intolerance is actually cause of this risk of death or just associated, but there's a clear association. And so all of these things related to sarcopenia contribute to significant public health costs. This is on top of the loss of quality of life. Obviously, you lose your independence, you can't function as well. It has a significant drain on sort of the psychosocial aspect of your life, but also these are just public health costs in dollars. can see, I think this is from the early 2000s. You can see it's the direct costs of sarcopenia are approaching $30 billion. And that's right behind heart failure. Now, of course, it's all dwarfed by diabetes. But there's interaction here. So this really represents a significant, or the age-related muscle dysfunction represents a significant cost both in quality of life and in dollars. So that's part of our motivation. That's what really drives us. And as I said, we're really interested in the mitochondria. I was interested in mitochondria before I was interested in skeletal muscle, and the two have kind of come together. So that's what we're going to talk about today, but I don't want to suggest that the mitochondria are the only cause of sarcopenia. This is clearly a multifactorial syndrome. But what's exciting for us, especially over the last few years, it's really becoming clear that the mitochondria are either a cause or a consequence of all of these other factors and many more that I haven't listed up here. So to us, that suggests that if we can have a significant impact on mitochondrial dysfunction, we have the capacity to make a real impact on quality of life, muscle performance, and public health. So we're coming at this from an oxidative stress perspective. And traditionally, when people think of mitochondria and oxidative stress, they think of this axis here, where you've got the mitochondria generating reactive oxygen species. That can lead to oxidative damage when this outstrips your oxidative defenses, or antioxidant defenses. This damage then leads to structural changes. That's irreversible changes in macromolecules. So those macromolecules have to be made new or turned over, leading to mitochondrial defects and ultimately muscle dysfunction. Now, probably, again, maybe over the last 10 years, really in the last 5 years, there's been a real appreciation for this other axis. And that's taking into account this idea of redox homeostasis or redox stress. So it starts the same way, that the mitochondria are a main source of reactive oxygen species, but instead of focusing on the damage, it focuses on how that affects the overall redox homeostasis of the cell. And so when you get excessive mitochondrial reactive oxygen species production, you alter redox homeostasis, and that affects redox signaling. And what I mean by this is mostly post-translational modifications. And what's interesting about this is that these are acutely reversible, and that they're in equilibrium with either the energetic or the redox homeostasis of the cell. Then ultimately, this can lead to inhibition and muscle dysfunction. So another interesting point about this is that this axis is really dependent on an interaction between the mitochondria and the cellular environment, because the mitochondria are controlling the redox environment, which then is feeding back on the mitochondria and affecting function. So this is kind of how we think about oxidative stress and disease in the lab. You're sort of under normal resting conditions. You're in homeostasis. You get some sort of stressor. This could be exercise, a high-fat meal, but it's within this range and leads to adaptive signaling. Now, if this stressor becomes chronic or exceeds some threshold, that can lead to a stress or an inhibition of function, then ultimately oxidative damage way down at this side of things. So we wanted to set up a system where we could study both aspects of this. And so as I mentioned, this relies on an intact communication between the mitochondria and the environment. So you have to look at the integrated system. So in order to do that, we're applying a technique we call metabolic spectroscopy. So this combines optical and NMR spectroscopy to measure oxygen consumption and ATP production by the mitochondria. So this is an image of our homemade probe. I guess probably don't need to tell you that it's homemade. The homemade probe and our 14T magnet at the UW. So we got the mouse body being held horizontal, and then the leg is pulled through the NMR coil, such that we're looking at the distal musculature. That's the gastrocnemius, the tibialis anterior. And then we flanked this NMR probe by fiber optic bundles, so we can collect optical spectroscopy through the leg as well. And so conceptually, it's really quite simple. And I should say that these are anesthetized animals, and they can fully recover. It's a non-invasive approach. So the approach is really conceptually simple in that we just really focus on what the mitochondria do, and that is they consume oxygen, where in the muscle it's bound to hemoglobin and myoglobin, and they use that to make ATP to meet the energy demand in the muscle. Now in the muscle, we have phosphocreatine, which buffers ATP through incretin kinase. So to get at the oxygen consumption side of things, we look at the optical spectra. So this is the optical spectra through a mouse leg under fully oxygenated or deoxygenated conditions. And we take advantage of the fact that the spectra for myoglobin and hemoglobin are different whether oxygen is bound or not. So we can induce some perturbation. In the cases I'm going to talk about today, it's going to be a brief ischemia. And then watch as the muscle goes from fully oxygenated to deoxygenated and use that to calculate the rate of oxygen consumption. For the phosphorus side of things, we just look at the 31P NMR. This is inorganic phosphate. This is phosphocreatine. And then these are the 3 ATP peaks. So as I mentioned, it's fortunate that we have phosphocreatine because this is like 25 to 30 millimolar, where this ATP is only 8 millimolar. And so something you'll notice here is that ATP is constant throughout this ischemia reperfusion cycle. Whereas once we make the muscle ischemic, mitochondria can no longer make ATP. We use this rate of, so PCR is broken down to meet that ATP demand. So that rate of PCR breakdown is equivalent to mitochondrial ATP production under resting conditions. So that gives us the ATP production. And so from that, we can get a P to O ratio, or a mitochondrial efficiency. Then we can also look at the rate of recovery of phosphocreatine, and that we can use that to calculate a maximum capacity for ATP production, or an ATP max. And I'm happy to talk more about this afterwards if anybody wants to, but I don't want to bore anybody too much with the details of it. So we first wanted to look at the effect of aging on these two parameters of mitochondrial function, so in vivo. So these are some data from Mike Siegel, who's a bioengineering grad student in the lab. They graduated a few years ago. So when we look at C57 black 6 mice, we go from 7-month to 27-month-old, we see a significant decline in the P to O ratio. This means that these mitochondria are less efficient. They're producing fewer ATP per oxygen consumed. And there's also a decline in their maximum capacity, so their ATP max declines. Now, so what we were really interested in is what role oxidative stress plays in this. So we treated these animals with a really low dose of paraquat to induce an oxidative stress. So paraquat's a redox cycler that steals electrons from complex 1 and generates superoxide. So this induces a mitochondrial oxidative stress, and what we see is that in the young animals, we can mimic the old phenotype as far as mitochondrial efficiency, and we see a further decline in the old guys. Same thing with ATP max. That it's a smaller effect in the young animals, but a significant decline in the older animals. So our mild acute oxidative stress is mimicking some of the effects of age on in vivo mitochondrial function. So our next question was whether this reflects oxidative damage or a change in mitochondrial content, loss of structure. I should say we made this measurement 24. We injected, and then 24 hours later we made the measurement. Okay. So to do that, we looked at permeabilized fibers. So for those of you not familiar with this, basically you take the muscle fiber out, permeabilize the sarcolemma. Basically, you wash out the cytoplasm. So at that point, it's kind of like an isolated mitochondrial prep, except they're still kind of contained within the myofibers. And then add various substrate inhibitor combinations to look at respiration. And when we did that, we found no effect on state 4 respiration of either age or paraquat. And state 4 respiration is a measure of proton leak. So in less efficient mitochondria, you'd expect elevated state 4 respiration. And here we see that there's no effect of either treatment, despite the different P to O ratios. And similar thing, no effect on maximum mitochondrial ATP production here.
How old are the old?
These old are 27 months old here. 27-month-old, and these are all going to be females, for the most part, black 6. So just briefly summarizing here, basically it's what I just said, that we can reproduce some of the aging deficits, and they don't appear to be reflected in ex vivo assays. So that led us to the conclusion that the environmental, or the interaction between the environment and the mitochondria are an important part of these mitochondrial deficits.
David, do you ever see actually an increase with age in oxygen consumption? For example, sometimes we see comparing 6 to 6-month-old to maybe a 18 or 20-month-old
Yes.
muscle, that actually there is a bit of an increase, and you have to go way out.
Yes.
Very late.
Yep. We do. And yeah, so we looked at the, just not to get too far off track here, but we did, I think it's 2015 in Aging Cell. Shane Cruz has a paper. We looked in the EDL versus the Soleus. And the EDL, we actually saw a slight increase. I don't think it was significant in respiration that we didn't see in the Soleus. We actually saw a decrease in the Soleus. And so we've seen that in a couple. Now, we actually just got some recent data that I'm not going to share today, or I'm not going to show today. I'm sharing it right now. We did see an elevation in the gastroc as well.
Yeah, we see it in the gastroc.
Which is, yeah, but then always, when you look in vivo, we see a decrease.
That's published?
The gastroc data is not published. The decrease in vivo is published, and the no change in the EDL is published, yeah. But I'm happy to talk more about that, because I think that's a fundamental part of what's going on. So this leads us to the going back here, that the cell environment is important, which I think goes to this point you're bringing up, that once you isolate those mitochondria, you're looking at the structure of the mitochondria. There may be more structure, but in their cellular environment, they're not working as well. So that may be the motivation to make more mitochondria, to have more structure. So this leads us to a hypothesis that maybe we can manipulate the redox state and we can see rapid changes in vivo energetics. So about this time, I started collaborating with Hazel Zetto and Peter Rabinovich, working with a mitochondrially targeted peptide that is now called elimipritide. Originally, or used to be called SS31, then it was MTP131, then it was Bendavia, now it's elimipritide. I'm going to call it SS31 for the rest of the talk. But that just by happenstance localizes to the mitochondria. This is a fluorescent analog. I think these are CaCO2 cells. This is a mitochondrial stain, and this is a co-localization. There's a lot of data on this showing the localization. It's also been shown to reduce mitochondrial H2O2 production. These are some data from Ethan Anderson and Daryl Newper's lab from rats fed a high-fat diet and then treated with increasing concentrations of SS31 and showing that the H2O2 production, or the mitochondrial oxidative stress, decreases. And it now looks like an important part of the mechanism for SS31 is its interaction with cardiolipin. So this plot shows another fluorescent analog of SS31 where the fluorescent tag changes its fluorescence, increases its intensity, and changes the spectrum in a hydrophobic environment. So when you incubate this with phosphatidylethanolamine or phosphatidylcholine, you don't see any effect where this is the control. But when you put it in the presence of cardiolipin, you see this strong change. And this is just really to remind me to bring up this point. There's a lot of data showing that there's this interaction between cardiolipin and SS31. And why is this important? So this is how we teach the electron transport system in the mitochondria. This is how we think about it a lot of times. Linear membrane with a linear chain of complexes. When in fact we know it actually looks like this. We have these tight curves, and the complexes form these super complex structures. Well, it turns out cardiolipin is this tetraacyl phospholipid that basically has the shape of a cone, and it facilitates the forming of these bends. Should get my notes here. Interacts with all of the electron transport chain complexes, and it's really thought to anchor these super complexes. So it's an important component to the inner mitochondrial membrane for efficient function of the mitochondria. It also happens to be preferentially oxidized under conditions of stress. And when that happens, it messes up this tight structure, disrupts supercomplex formation, disrupts the curvature, and the hypothesis is it makes the mitochondria less efficient. So armed with the localization and this effect on this effect of the peptide on hydrogen peroxide production in other models, we wanted to see what effect it had on H2O2 production in young and old muscle. So this is H2O2 production. It's the Amplex Red assay in the EDL muscle from young and old animals. And these are paired assays where one leg is done without and one leg is done in the presence of SS31. So the first thing you'll notice is a significant increase in hydrogen peroxide production with age. There's a lot of variation. But when done in the presence of SS31, this is significantly reduced.
This is an isolated mitochondrial permeable isolate.
permeabilized fibers. Yeah. Have we done this? We have not done an isolated mitochondria. Other people have done an isolated mitochondria, but we haven't done it in old. We don't do much isolated mitochondria work. We mostly do permeabilized fibers. So another take-home is that it looks like the more dysfunction of the mitochondria, the greater effect of the treatment. And in fact, the young animals don't appear to be affected at all, and the old functional ones don't appear to be affected. So this is telling us two things. One, it doesn't affect healthy mitochondria. And two, it's not a scavenger, right? Because if it were a scavenger, it would look like this. When this assay is done in the presence of catalase, all these would go to zero because your hydrogen peroxide would be scavenged. Okay? And just to, because this was hotly contested for a while, so we just did a further experiment. This is a biology-free system where this is just looking at hydrogen peroxide in the presence of Amplex red and in the presence of two different concentrations of SS31. You can see that there's no effect of the peptide on this hydrogen peroxide signal. So armed with this reduction in oxidative stress in the old mitochondria, we set up a really simple experiment. And again, this is Mike Siegel's work. We took young and old female black 6 mice from NIA, injected them with SS31, and then an hour later measured in vivo energetics, muscle fatigue, and redox status. And we published this a few years ago, so I'm going to kind of zip through it. But basically what we saw was a restoration of the redox status, the glutathione redox status, a reversal of mitochondrial inefficiency. So this is the decline with age. This is after an hour of treatment. This is in vivo mitochondrial function. An increase in the maximum capacity for ATP production after an hour. And then in a completely separate set of experiments, because I didn't believe it when he actually brought these data back, we look at muscle fatigue. So this is in situ preparation of a tibialis anterior, and this is repeated tetanic stimulations, I think every 2 seconds for 4 minutes. These are the old animals treated with the saline, and these are the old animals treated with SS31. So associated with the improved energetics and reduced redox status, we see increased fatigue resistance. And then to look at the question that you were asking about earlier, if we do look at an ex vivo system, so we take these same animals, they've been treated, then we look at permeabilized fibers. We don't see any effect on state 4 respiration of age or treatment or on maximum state 3 respiration. And no effect on electron transport chain protein expression. So we're not remodeling anything. We're not creating new structure. We're not creating new structural capacity. We're just manipulating function. So, like I said, we published that a few years ago, and then since then, we've been trying to focus on longer-term treatments. And so this is some data. These are some data from Matt Campbell in the lab where we did an 8-week treatment now. And now the delivery is by osmotic mini pump. So this is a continuous infusion over 8 weeks. We do a baseline endurance capacity on a treadmill, treat them for 4 weeks, switch the pump out, another 4 weeks, and basically do the same thing, measure in vivo energetics, fatigue, and exercise tolerance. And same thing. Reversal of the mitochondrial deficits. Increased P to O, increased ATP max. Now in this case, we also saw an increase in resting levels of ATP, which we didn't see under acute treatments, which we wouldn't expect to because ATP levels in the muscle are really tightly regulated and don't change acutely generally.
That's measured in the biochemically or in the biochemically?
This is biochemically. Yeah, so this is an homogeneous with HPLC. And we also saw improved muscle fatigue or fatigue resistance. These are the old animals up here, treated, untreated. These are the young animals. No significant effect on fatigue resistance, although there's something interesting there that we haven't pursued. But no effect on specific force. So the force generating potential of the muscle isn't different, but its fatigue resistance is. Now, I think Peter Rabinovich was out here last year. And so we're doing a lot of these experiments in parallel to Peter. So he's looking at the cardiac side of things. We're looking at the muscle side of things. Excuse me. So these are some data from Ann Chow. So I'll just go through this pretty quickly because I'm not a cardiologist and because I think a lot of you have probably seen it recently. But if she looks at old animals treated for 8 weeks with SS31, she also sees improved diastolic dysfunction. This is the E over the A ratio. That's the passive versus active filling. So it's kind of a measure of the ability of the ventricle to relax. This is the Young level. The active filling of the ventricle becomes a greater component, so this ratio decreases. After 8 weeks of treatment, this significantly improves. The myocardial performance index is kind of a composite measure of both the systolic and diastolic function. And there's a trend towards improvement, but there's no change in fractional shortening. But there is just appear to be a reversal, or at least or maybe a prevention of hypertrophy in these old animals. And I should note that these are male mice. The data that I've been showing have been for female mice. So we aren't seeing a sex-specific effect. So not surprising if we see improved muscle function and improved cardiac function, we see improved exercise tolerance. So this is treadmill running capacity. These guys are running at 30 meters a minute for 10 minutes. These are the baseline measurements, and at 8 weeks. These are the young guys up here. You can notice the broken axis. So the young animals run quite a bit more than the old. But what you clearly notice, you don't really need statistics for this, is that after 8 weeks, the untreated animals don't really change, but the treated animals significantly improve their exercise tolerance. And that, if you look at the delta plot, you can really see that here. Now, one thing that was particularly interesting to us, which I didn't really expect, was we also saw an increase in the gastrocnemius mass after 8 weeks of treatment. So this is the gastroc normalized to tibia length. So it looks like even though we're starting this treatment at 26 months old, we are able to either reverse or prevent further decline that allows us to have this slight rescue of muscle mass. So I think I've basically summarized these things already. We get a reduced mitochondrial oxidative stress. And 1 hour and 8 week treatments are both improving energetic and muscle function, but no effect in healthy mitochondria. Yep.
Do you know what it does to AMPK?
Maybe. So yes, so we have some data that I'm not going to show just in the interest of time, and it's kind of off track, but we're really interested in that now, how the mitochondrial oxidative stress may be affecting cell signaling in the muscle. So preliminarily, we are seeing that there's an elevated phosphorylation of AMPK in the animals treated after an acute treatment in the old animals. So we get a depression of phosphorylated AMPK at rest with age. That then looks like it is bumped back up with treatment. We have some ideas, but we don't really know a mechanism for that. Because you think it would be the opposite. If you were storing energy, reducing energy stress, restoring mitochondrial function, your energy stress signals should go down. But I think it may be a complicated interaction. Yep. Yeah. Well, at its simplest level, increase energy supply. If we're getting increase in ATP max, so the mitochondria have a bigger reserve, they can make more ATP and maintain muscle function longer. That may be an oversimplification. As I'll show a little bit later, we're starting to really get interested in the interaction between the redox status, the energy status, and post-translational modifications to different parts of the muscle. And there are some differences in myofibrillar and EC coupling proteins. But right now, that's kind of wild speculation that may be contributing to that. But the simplest answer, and the one that we talked about in the paper that we wrote for the acute stuff, was just that you have more mitochondria, better mitochondrial function, you're more fatigue resistant. Yeah.
So how do you deliver the FX31 to the mice?
So for the acute experiment, the 1 hour experiment, that's an IP injection. For the 8 week experiment, it's in an osmotic mini pump that's put subcutaneously.
Okay, because I wonder if it's a direct effect or indirect effect on the muscle. It could be a systemic effect, right? Not through.
Yeah. Yes, absolutely. It could be, you're improving, cardiac function, and that's leading to improved muscle function, or is that what you mean?
Yes.
Yeah.
So do you have any evidence showing that the SS31 actually going to the skeletal muscle mitochondria?
Yeah. Well, we have evidence showing that it goes into the skeletal muscle. We haven't done the localization experiments to show that it's actually associated with the mitochondria in the skeletal muscle. Right. Yeah. Yeah. When we've tried to do that, and it's been a challenge to, yes, but we thought the same thing, but we haven't been able to do it. Yeah.
So, since I'm assuming the design was of the same different metabolism of cardiac muscle and skeletal muscle, one more dependent on lipid metabolism, which is an entirely different set of processes, produces a huge amount more ATP. And then you've got skeletal muscle, which is working primarily in glucose.
I don't know under resting conditions if it is.
So, when you sit with the gastroc, you actually saw muscle hypertrophy. It got bigger.
Yeah.
So, when you're giving the SS31 over 4 to 8 weeks, or whatever the time frame is, you see cardiac muscle hypertrophy.
We see the opposite. We see a reversal of the pathological hypertrophy.
So, old rats have pathological hypertrophy.
Old mice, yeah. Yes, they do.
In general, and that's reversed by SS.
Yes. Right. Yeah. Yeah. So this is kind of the incomplete part of the story now. Now we're trying to understand the mechanism, and that is now the mechanism of once you improve mitochondrial function, how does that translate to improve muscle, and how is the mitochondria improving? So first, we're going to talk a little bit about this side of things, and these are the structural changes. So this is these are things that are going to travel with the mitochondria. Restoration of or replacement of oxidative damage, mitochondrial protein expression. So the first thing we saw, which was interesting, is if we look at protein expression in the electron transport system. So these are subunits of all the complexes of oxidative phosphorylation. You can see going from young to old, in many of them, complex 1 particularly, but also complex 3, and to a less extent, 4 and 5, we see an increased expression. No effect on complex 2, but then after 8 weeks of treatment, this is reversed. So it's renormalizing or restoring this protein stoichiometry. And in that same aging cell paper I was referencing earlier, we see a similar thing with the MCAT, in that with age we see an elevation of some components of the electron transport system, particularly complex 1, and then in the MCAT mice, you don't see that. So you get protection from that. What we're referring to right now is sort of aberrant protein expression or aberrant assembly of the electron transport system. We're also seeing a restoration of the redox status. So this is reduced glutathione, oxidized glutathione, and the ratio there. This is after 8 weeks treatment in the gastroc. And reversal of oxidative damage. All this supporting the idea that reducing mitochondrial oxidative stress is associated with the improved energetics and improved function. So this is an H&E Western blot, just to fully describe this. Now, I mentioned earlier that cardiolipin is one of the targets of SS31, like basically the identified target of SS31. This is this conical structure, important for forming these bends in the membrane. And when it's oxidized, it goes through this remodeling process and goes through this 3-acyl chain step where you get, it's called the monolysyl cardiolipin, which reduces its ability to form these bends, reduces its interaction with the complexes. So we expected to see a restoration of cardiolipin content in the skeletal muscle. Now, we didn't see any effect on total cardiolipin. And we also didn't see any effect on isoform. I was talking to someone earlier about isoform expression. Basically, the isoforms don't appear to change with age, at least in our measure. This is Zenlin Han at the Sanford Burnham made these measurements for us. So we don't see any change with age, no effect of treatment. Yeah. Because if you had more mitochondria, if you have a change in mitochondrial content, you'd expect there'd be more cardiolipin, but it wouldn't tell you anything about the quality of the mitochondria. Yeah. But when we looked at this intermediate or this immature form of cardiolipin, we see a significant increase in going from young to old. So we're interpreting this as increased damage or increased remodeling of cardiolipin that is partially reversed in the treated animals with age. So another aspect of mitochondrial structure that appears to be improved by this 8 weeks of treatment. So this is basically what I said here. So, but this explains what happens after 8 weeks, but it doesn't really tell us what's going to happen after, how we're getting these treatments after an hour. So for that side, we're focused more on this sort of dynamic sort of redox change thing. So to do that, we got interested in thinking about the thiol proteome. So thiols are contributed to proteins by cysteines. Under reduced conditions, you have this sulfhydryl group. When it's exposed to hydrogen peroxide, it forms a sulfenic acid, and then it can interact with glutathione and form a S-glutathionylated adduct or modification. It can also be sulfhydrated or nitrosothiolated. Now, what's important is that all of these in blue are rapidly reversible. These are essentially in equilibrium with the glutathione redox status. Or it can be further oxidized to sulfinic or sulfonic acids, and these are examples of oxidative damage that are irreversible. Basically, you have to break the protein down and make new protein. And as I got into the literature a little bit more, I got more and more interested in this side of things as I looked at mitochondrial proteins that were actually regulated by S-glutathione. So these are actually just a few of them. I've run across several more. But complex 1, complex 4, this is the ATPase complex 5, several aspects of the TCA cycle, as well as the many redox proteins that I haven't outlined here. So this suggested to us that this may be a mechanism that you could get these rapid changes in your in vivo energetics that might be lost when you isolate the mitochondria and you normalize the redox environment. And so as I started getting into this area, the thing I learned about redox chemistry is that I don't want to do redox chemistry. I want to find someone that knows what they're doing. So we teamed up with Wai-Jun Qian at the Pacific Northwest National Lab, who's developing a redox proteomic approach. And the key for any sort of redox measurements, as many of you probably know, is a rapid blocking of any reactive thiols so it doesn't oxidize while you're processing it. So the first step is you take your homogenate, treat it with NEM, which binds to any reduced sulfhydryls. Then, so this is a reduced one. It's blocked by NEM. And then these are modified ones that aren't altered by the NEM. Then you can go through a selective reduction. In this case, we use glutaredoxin to focus in on S-glutathione. And then you do basically a pull-down or a trap column, and then digest and do your thioproteomics. Basically, the take-home for all of this is the 8-week treatment with SS31 reverses the age-related oxidation of the thiol proteome. So there are two comparisons up here. There's a young versus old and an old versus old treated with SS. Blue indicates sites that are less glutathionylated, and red indicates sites that are more glutathionylated. So we can see the old all clustered together to the exclusion of the old treated and the young. So basically, the treating for 8 weeks makes the thiol redox proteome of an old animal look more like a young animal. And these are all of the proteins that were significantly different in both comparisons. You can see across the board, we see a reversal of this effect. And I've just highlighted some of the ones that we're pursuing that were of particular interest. And then if you look at the whole data set, the real take-home message to us was that you see these modifications throughout the whole proteome of the muscle. So this is the old-young comparison, and this is a GO analysis. So this blue pie are proteins that were significantly more oxidized in the old animals that are associated with myofibrillar or EC coupling. These are mitochondrial proteins, glycolytic, and these are redox or protein quality control. So you can see that although we're targeting the mitochondria, the redox status, we're having an effect throughout the muscle. And similarly, we're not targeting up here. This is just going from old to young. Got ahead of myself. Down here, when we're specifically targeting the muscle with the SS31 treatment, we're actually seeing reversal in these same compartments. So myofibrillar, mitochondrial, glycolytic, redox, and protein quality control. So by manipulating just the mitochondrial redox status, we're having an effect throughout the muscle that could explain muscle performance independent of energetics or ATP production. And that's something that we're actively pursuing now, and it's something that's kind of an exciting part of the lab. So that's basically what I'm trying to highlight here. So going back to the idea of mitochondrial deficits or mitochondrial defects with aging, it looks like there's really sort of two components or two phases of this. And so if we just use ATP max per some measure of mitochondrial content, citrate synthase activity, as an index of mitochondrial quality, this is what we get in the young animals. In the old animals, we see a significant decline in mitochondrial quality. When we do...
I'm sorry, this index is, again, ex vivo.
This is in vivo.
Well, that's right.
Yeah. The ATP max is in vivo. It's kind of a combined thing. So it's an in vivo measure of function and an ex vivo or homogeneous measure of structure or content. So you can think of it as a function versus structure. So function decreases for the given amount of structure, we get poor quality. Then we do an acute treatment, manipulate mitochondrial redox status or mitochondrial oxidative stress, and we see an improvement in mitochondrial quality. Then we do a longer treatment, and we see a further improvement in mitochondrial quality.
Maybe this CS activity decrease with a model?
No, it does not. And so we generally have measured it in 26 to 28-month-old animals. I have a feeling, based on your comment earlier, you probably agree that if you looked at 32-month-old animals, it probably would decrease.
We looked at up to about 24, 28, and we don't see it.
Yeah.
That's why I'm asking.
Yeah, no. If anything, it goes up in some situations. And there was some data. There was a paper published a couple years ago in rats from Russ Heppel's group, where basically they didn't interpret it this way, but their data clearly showed that they were seeing the same thing. They were seeing an elevation in mitochondrial content in their older animals. So that's what's leading to this reduced quality. More content, less function. You get this partial increase in function, but you're not changing structure or content here because this is a rapid treatment. But after 8 weeks, you're getting less structure, better function, so you see further improved quality. And that's basically what I'm highlighting here is that, so this we're suggesting might be this dynamic post-translational modification of the energetics, whereas with a prolonged treatment, that restores quality by improving structure. And basically that's kind of what I'm showing here. So this is an improvement that can happen on the stage of, or the range of minutes to hours, where this maybe days of weeks with a treatment or exercise training or something like that, all leading to improved muscle function. Now, so the last few minutes, I'm going to talk about some work that's really not my own, but when I came across this paper, it made me really think about this. So this was in the, I think it was the most recent Science. This is kind of a where are they now kind of thing for the, the wonder molecules. You can see a lot of ones we would probably all recognize up here. Some of them have panned out to a greater extent than others, right? So, we showed a lot of really interesting data, and we're very excited about these results in a mouse model. But the question is, how does this translate? It's one thing to cure sarcopenia, improve sarcopenia in mice, but no one's going to really care that much. So I'm going to share a little bit of preliminary data from 2 clinical trials going on with, now I'm going to use elamipretide, because now we're in the clinic, right? For myopathy, yeah.
Before you go on with
Yeah.
the mouse study, do you have any feeling if the reversal, particularly the long-term reversal,
Yeah, yeah.
It's a lot longer experiment.
No, we haven't done that yet. But if the PO1 is renewed, that's one of the experiments we'll be doing. Actually, and I know Peter's lab is doing that for cardiac function, and they are seeing some persistence. I don't want to get into his data too much, but they are seeing some persistence. But it's not. It's not a one-time dose thing, and you're set for life. You definitely it reverts.
But it's not necessarily that SS31 has to be there to modulate the Thiols or whatever else.
Correct. Correct. That's what it suggests. Now it could be in the case of if you get a longer-term treatment where you replace your structure, you improve your structure, you're kind of starting over again, and then you might maybe decline again. You know what I mean? So maybe you're not quite seeing the same benefit, but you're improving things to the point where now you have to get the slow decline again. Yeah. Right. Okay. So quickly we'll go through a few. So this is a first the disclosure. These were two studies funded by Stealth that I was peripherally involved in this one just because it was based on our acute mouse model experiment. I had nothing to do with this one at all. I'm just sharing some, I think, interesting data. So this first one has the nice acronym of SPIT-M. I'm not sure who came up with that, but so this was PI is Bob Roshan-Robin, and Kevin Conley was the main investigator on this, making similar measurements to what we're making in the mice, the in vivo energetics in humans. So these were elderly people were screened for mitochondrial dysfunction, low ATP max, low P to O. They got a single dose of elamiprotide, remember it's SS31, infused over 2 hours, and then immediately taken down to the magnet to measure mitochondrial function. So they were screened beforehand, so you had a baseline, and then measured immediately after infusion, and then also 1 week after treatment. So I'm only going to talk about the ATP max data because these are under development. But this is the screen population. This is measuring this muscle, this flexor digitorum interosseous muscle. This is this muscle that moves the finger like this. And these are some data from Kevin's lab from an earlier study, just showing this is the vastus lateralis in the leg, just showing the range. So you can see there's a huge range in the old population. There's some old people that are pretty healthy. Right? So this is the group that were included in the study here. And then they either got a placebo or a 2-hour infusion of SS31. So this is the change in mitochondrial capacity, either immediately after infusion or 7 days later. This is the placebo group. So this is probably about, I think they said this is about a 5 to 10% increase. This is the elamiprotide infused group. This is immediately after infusion, significant elevation in maximum ATP production, ATP max, that then returns to, heads towards baseline 7 days later. So this was exciting for us because it really kind of validated the stuff we did in mice, and basically showing that this single dose has a significant effect on the energetics. Now, this other study is in a genetic mitochondrial disease. So these are the investigators. They're kind of scattered around the country. Again, I had no role in this, but this is the study design. It's a dose escalation study. 0.01 milligrams per kilogram, 0.1 milligrams per kilogram per hour, or 0.25 in infusion for 2 hours. And for each cohort, there were 9 treated and 3 placebos. The study design was a little more complicated. They did a baseline 6-minute walk test, then 5 days of infusion, and then a 6-minute walk test at 5 days, and then at 7 days. I'm only going to show 5-day data here because those are the only data that I have. I think these other ones are still being analyzed. But when you look at the distance walked, this is the change in distance walked. At the highest dose of treatment, you see a significant improvement in their muscle function here, their exercise capacity. In these, now, I forgot to point out that these are adults. These are 16 to 65-year-olds. So these aren't kids with mitochondrial genetic diseases, but they're older people. So what was really interesting to us is then when the investigators broke this down a little more and looked at a model to relate the effect of the treatment versus the baseline, what they saw basically coincided with what we saw in the animals, is that the healthy people weren't really affected that much. They weren't improved. So this is the baseline distance walked in 6 minutes. And these are people that are doing pretty well. I'm not familiar with what's expected in this, but I'm told up here is kind of a normal range. Okay? There's no effect of any of the doses. Now, down here, these are very dysfunctional people, and they see the greatest effect, and there's a significantly, I guess, negative slope associated with the greater dysfunction, the greater effect. So both of these studies, we were kind of waiting with bated breath to see these results, just to see, the idea that actually something, actually translated was pretty exciting for us. So I kind of jumped around a little bit and kind of took you through sort of the journey we've been on in the lab, but basically the take-home messages are, one, that there are these two aspects of mitochondrial function. You have this structural component that you're going to see when you isolate mitochondria and permeabilize fibers, and there's also this dynamic aspect that looks like it's dependent on the integrated system and the role of the mitochondria in the cell, and that SS31 can reverse skeletal and cardiac muscle dysfunction, and that by manipulating the redox stress, you can manipulate the redox-dependent protein post-translational modifications throughout the muscle that can have effects independent of energetics. And we're actually now starting to look at the phosphoprotein as well to see if we see a similar effect. And that this may be an interesting and important strategy to improve quality of life in the elderly. And so I want to just thank members, past and present, and members of the lab. Mike Siegel did a lot of the acute stuff in paraquat. Matt is doing the 8-week treatment. And Phil is doing some really nice signaling stuff, the AMPK data that I talked about. Nathan Schoch Center, collaborators, especially Kevin Conley and Hazel and Waijun for the Thiol Proteomics. Stealth gave us the peptide. And then all the people that paid the bills here. And that's all I have, except I want to make a plug that we will have at least one postdoc opening in the lab in the spring. And next spring, there are several openings at the Aging Training Grant at the UW for postdoctoral positions. So anybody finishing up in that time span, interested in moving to Seattle and seeing Mount Rainier and doing some really nice aging work, let us know. All right. And I'll take some questions now. We have some time. Yeah, someone else asked me that earlier. Yeah. Depends how you want to live. Probably not as well as you can live down here.
Only take pellets?
What's that? What?
You only take pellets?
Oh, yeah. Right. Yeah, yeah, that's definitely something to consider. Yeah. Question? Yeah. We have not seen any toxicity, and has been reported to me by the company in their phase 1 studies. There's a really big tolerable range, order of magnitude beyond what we're treating with. And I think it's from our experiments, it's kind of a nice system in that it doesn't appear to affect healthy mitochondria, which. And I think an important point is something we always try to make in our NIH grants is that it's not an antioxidant. It's not a scavenger. We're overrun with scavengers that have not held up. Because when you scavenge, you're kind of indiscriminately screwing up redox signaling. Whereas what we think is happening here is if we're restoring mitochondrial efficiency back to a normal level, you're not going to interfere with normal redox signaling. That's the hypothesis.
What about the bioavailability? Accumulating muscle?
From the data that I've seen, so there's a relationship between mitochondrial content of a tissue and its tissue uptake.
So the tissues reaching mitochondria tend to have more of a SS31.
Right. But the pharmacokinetics are really fast. So by 24 hours, I think 90% of it is excreted in the urine. So it doesn't appear to accumulate. It localizes, but then doesn't accumulate.
So SS31 is related to that mitochondrial heteroplasm?
Yeah. So what's the question? What's the first part?
SS31 can be related
to heteroplasmy. In the sense that if you have heteroplasmic mitochondria, you have some population that is dysfunctional and some that are working normally. I guess the prediction would be the ones that are dysfunctional. You would get improved function if the dysfunction had to do with a sort of disruption of the membrane structure. I think it probably would only be indirect. Because I would think that the heteroplasmy might induce a more oxidized state, which then kind of would affect throughout the mitochondria. But I guess for localized, yes. Yeah, it's kind of a wishy-washy answer, but I'm not really sure.
Do you have any idea what the mechanism is for the Cardiolipin modeling? Because SS31 itself doesn't do the modeling.
Correct.
Does it bring in one of the Cardiolipin?
Yeah. No. So no, that's a really good question. What we're thinking, the model I have in my head, is that basically what you're doing is the stressor that was causing the remodeling was the oxidative stress. And if you're reducing that oxidative stress, then you're allowing sort of the natural remodeling to kind of catch up and reduce stress. Again, that's the model I have.
So not necessarily boosting any of the remodeling systems, but just
Right.
letting them
Right. Yeah, we've had. So I think I've talked. We measured. Well, as Hazel in collaboration with us, Hazel Zetto measured in collaboration with us tafazin expression. Didn't see any change in tafazin expression or anything in our treated muscle. But that's all we've done. We haven't looked at the cardiolipin metabolic pathway to any extent. Yeah.
Please pardon me if you can actually answer this already or show data. What's the state of glutathione in myocytes? Is it restored magnesium equivalents? Is that where the
Yes. Yeah. So in both cases, both experiments, we see with age we see a reduction in reduced glutathione that's restored after an hour treatment and then also after the 8-week treatment. Yep. I think there's a question over here. No.
I want to know if you have tested after treatment, is there any supercomplex change?
Yeah, that's we would really like to know that, and we just recently isolated the mitochondria to do those experiments. Yeah, and again, if we get, we'll get it done a lot faster if we get our PO1 renewed. But either way, we're in the process of trying to do those experiments. Because that's kind of the hypothesis, right? That you're restoring structure and making the electron transfer more efficient. So we'll see. Yep.
The SS31 shows same effect with the catalyst transgenic, right?
Oh, so we have not done the experiment, looked at skeletal muscle function in the MCAT mice. I know the Rabinovich lab has looked at the MCAT and the SS31, and they are not. You got the MCAT effect, you got the SS31 effect, and you put the MCAT and the SS together, and you have something different. And that's all the detail I can really tell you. That's all I can remember.
Tell you whether it's really the loss having caused that, right? It's the function to
right. Yes, there were some complications with those experiments, I think, but I think that was their motivation for doing those. And
it's not very conclusive.
It's not. I think inconclusive is the best way you could say that. Yeah, you cannot completely rule out that there's something else going on with this, and we haven't ruled that out. This is the mechanism that we know is happening. There may be other things going on that we haven't been able to identify, and we're pursuing some other pathways, some more discovery type pathways to try to figure out other potential targets. Yep.
So, do you think this compound will affect lifespan? It's not inhibition mitochondria doesn't mean normal lifespan.
Right.
Will you
I wildly speculate on that. I would think initially going in, I would say yes because of the MCAT results. But based on my answer to this question, you might think, well, that doesn't make sense. So I do think it would. We've been trying to do that experiment. So we've just started an experiment where we started treating at 20 months, and we're doing weekly injections, and we're going to follow them out. It's not a lifespan experiment. It's a function experiment. We're hoping to do the lifespan experiment, but yeah, that is a good question.
Has the effect of SS1 on the distal morphology been looked at?
Yes. In the heart, it's inconclusive, but it's not as dramatic as you might think. In the skeletal muscle, see, one thing with aging skeletal muscle in mice, there's people that have worked with aging skeletal muscle in mice, the muscle doesn't look that bad. You don't see these swollen mitochondria that you might expect, all dysfunctional, big vacuous balloons. They look pretty good. And in our very, very preliminary look at it, we didn't see an effect. People have looked at it in kidney, I think kidney and maybe heart too, ischemia reperfusion models. And that does appear to improve or protect cristae function. But in the cardiac and the aging skeletal muscle, I would say the jury is still out. And I'm not really anxious to do those experiments just because in order to do EM well, it's really tedious. So we focus more on the functional aspect. But if we see super complex or we don't see super complex differences, I think at some point we're probably going to have to do it. Did you have a question?
Yeah, going back to what Adam said, cardiolipin oxidation has been shown to link to apoptosis, to that cytochrome C nexus. So I'm thinking if there's type of mitochondria, so that mitochondrial turnover, how much of a role does it play in your improved mitochondrial function?
Oh, yeah. We in this model, we have not measured atrophy or, autophagy to see if that gets stimulated. And we have not done turnover measurements in the skeletal. We've done turnover measurements in the multiple. No, we haven't done any turnover measurements in this model. So I don't know if I don't know if it's we're just fixing mitochondria that are there, or it's basically, like you said, cleaning house, the spring cleaning, just kind of getting rid of all the crappy stuff. I don't know. Good question. All right. Thank you. Thanks, Dan, for your attention.